love watching people do it because it's really. Alright, Connie, make sure I do it too. Alright. It's like his virtual. Yeah.
You don't want to
hand out the PowerPoint, calibrate the board, and then we're ready to start.
Alright. There you go, pass this around.
You don't want to put that board on the top.
Alright, so I actually wanted to go back a little bit because I found a better video that explains pressure flow. Alright, so last time we talked about plant tissue. So we talked about ground tissue, dermal tissue, and vascular tissue. We talked about the 5 kinds of plant cells, which are, what are they? Parenchyma, xylem cells, which are, what are the 2 xylem cells? Tracheids and vessel elements, and then
Tracheids and what?
Vessel elements?
Vessel elements.
Vessel elements, yeah. Tracheids and vessel elements. And phloem cells, which are called sieve tube members. And what is it that sieve tube members don't have?
What?
Sieve tube members are missing something. What are they missing? They're missing a nucleus, remember? Talk about that? And I talked about it in a couple of classes. So yeah, so sieve tube members, the phloem cells, don't have a nucleus. They're alive at functional maturity, unlike vessel elements and tracheids, but they don't have a nucleus. Why do you think a phloem cell wouldn't have a nucleus?
More space.
Yeah, it's the same reason a red blood cell doesn't have a nucleus, right? More space. So it allows them to serve more space. In fact, over the course of the phloem cell's life, a lot of their organelles actually wind up degrading. They get sort of pushed to the side and they degrade. And it turns out phloem cells don't, even though they're alive when they're working, they don't live for very long. They live about a year or so. Maybe more in some plants, but about a year. All right, so last time we had talked about transpiration. All right, and I showed you the David Attenborough video, yeah? Okay, and transpiration, what are the properties of water that allow transpiration to happen?
Adhesion.
Adhesion and cohesion, yeah. The ability of water to evaporate is what allows that transpiration. But it's adhesion and cohesion that pull the water up the plant. And there's another thing that can affect it a little bit called root pressure. The water is kind of pushing to get into the roots. The roots have a lot of solutes in the cells of the roots. So water is trying to push in a little bit, but it's not nearly enough force to drive water all the way up the tree.
So how do these trees, like, you know how like when you watch National Geographic in Africa, they're like, how do those work? Like, because there's not enough water and it's like in the desert.
Oh no, they're still getting groundwater.
Oh.
There still has to be enough water somewhere down in there, down under the surface, for them to get water. Yeah, so most trees transpire quite a lot, and plus, desert adapted plants have, like, for example, conifers out here have those waxy, thin leaves. Right? Their needles are actually their leaves, and that waxy coating helps prevent some water loss. The low surface area of those leaves also has to prevent some water loss. And then you get things like can plants and C4 plants that have ways of keeping their stomata closed during either the whole day or at least dry parts of the day. Okay, so last time we were talking about pressure flow. So we got through transpiration, which is how xylem goes. Now we're talking pressure flow, which is how sugar moves. So the basic idea is, okay, I don't know where that came from. The basic idea is, where's my cup of, there. All right, if Walter over there is xylem. Walter has water in him, yeah? Because water goes up the xylem. If Julie here is a phloem cell, and I'm a photosynthetic cell, so I'm making sugar, okay? I make sugar. The sugar gets loaded, usually via active transport. The sugar gets loaded into Julie the phloem. Now Julie the phloem has a high solute concentration. So where do you think that water is going to go? Right. Okay. Now, once Julie is full of water, she's kind of like a water balloon, right? She's been filled with water. So what's the pressure like on the walls of a water balloon that's filled with water? Yeah, right. So she's actually got high turgor pressure, right? There's a high psi P in Julie now. Okay, water squished in her. So the water's now going to squish out of Julie down the phloem until that phloem gets to a place where there's a need for sugar. Okay, so over here, Stephen's busy becoming part of a fruit. Right, so he needs to build up lots of sugar. He's a cell in a growth fruit. So the sugar leaves the phloem over here. Now, what's the solute concentration in the phloem now? It's low. Okay, so it turns out now there's no low solute concentration. So now that water can osmose out of the phloem through the plant, and a lot of it actually winds up back in the xylem. Okay, so that's the basic upshot of pressure flow. We load up the phloem with sugar. Water follows, chasing that high solute concentration. Lots of water now in this phloem cell makes the cell really, really turgid, like full of water pressure. So that water squishes out of that phloem cell, squishes through the phloem until it gets to a place where the sugar in that water is needed. The sugar is then moved out, either actively or passively, depends on what kind of cells we're talking about. Moves out either actively or passively into a cell that needs the sugar. Say part of the fruit, say a root cell that can't make its own sugar. Somewhere where we need that sugar. Because now the solute is lost in the water potential in that phloem is down. So now that water actually can osmose out and up. So that water can now osmose out into areas that have higher solute concentration. A lot of that water actually comes out. So let's watch a little clip and then we'll go on. I didn't do a good job of covering it last time I had you guys, so I wanted to go back over it. Oh, let's hook up the sound.
Tissues called xylem and phloem. Xylem transports water and minerals, while phloem transports a variety of dissolved substances, including sugars and amino acids, throughout the plant. Water in the xylem always moves up in the direction from the roots to the leaves. At the leaves, water evaporates. This evaporation, called transpiration, creates tension at the top of the water column in the xylem and causes water to be drawn upward from the roots. Unlike in the xylem, the solution in the phloem can flow up or down the plant. The direction depends on the concentration of solutes in the phloem. A model called the pressure flow model describes how sucrose concentrations determine the direction of the flow. Two types of cells, source cells and sink cells, play roles in the transport of solutions.
Okay, so just a couple quick things. So phloem can go up, all right? So if you're a leaf down here, you make a lot of sugar, and you're an apple tree, and the apple tree is putting an apple out up here, sugar can actually go up because it's pressure that's driving it. Okay, it's not, it's not just gravity, it's actually pressure that can push it up or push it down. The other thing is this, he calls it two types of cells, source cells and sink cells. That's really a descriptive term. It just means cells that are making sugar versus cells that aren't. It's not like if you open up a plant, you can say, oh, that's a source cell. Like you could say that's a parenchyma cell. It's not the same thing. A source cell is any cell that's making sugar. A sink cell is any cell that needs it.
In the phloem. Source cells produce sucrose and load it into the phloem. Source cells are typically photosynthetic leaf cells, but they can also be other cell types. For example, root cells with large stores of carbohydrates can act as source cells by releasing these carbohydrates into the phloem. Sink cells are any cells that unload sucrose from the phloem. Sink cells are found throughout the plant, but are abundant in roots or developing fruits and shoot tips. Sink cells typically cannot meet their own needs for carbohydrates and must import them from the phloem. The fluid begins to flow when source cells pump sucrose into the phloem. As the concentration of sucrose in the phloem increases, water enters the sieve tubes by osmosis. The water enters from any surrounding tissues, such as the xylem, that have lower solute concentrations than the phloem. As water flows into the sieve tubes, the turgor pressure within the sieve tubes increases. This pressure forces the sugar solution through the sieve tubes to regions of lower pressure. The solution passes by sink cells, and these cells take up sucrose. As the sucrose concentration decreases, water flows by osmosis out of the phloem to regions of higher solute concentrations. The loss of water, in turn, decreases the pressure in the phloem and pulls fluid from other higher pressure regions of the phloem.
Okay, so does that make sense? Okay, I think that was a better video than the last one I showed you. So why I wanted to go back. All right, come on. There we go. All right.
Does the xylem only operate in one direction?
The xylem operates, yeah, because water is only coming in through the roof.
So it's only going up?
And it's transpiring out the leaf. So it's only going up, yeah. Okay, so we were doing this, we had gone through plant nutrients. All right, and we had finished when we were talking about the layers of the soil. So it's helpful both for ecology and plant biology to have a little bit of an understanding of soil. We don't need to go into a whole lot of detail. But again, we talked about how the humus, which is or humus, the stuff on the top, is decaying organic material. And I've heard it said both ways. Sometimes that's called the O for organic horizon. Under that, there's the topsoil or the A horizon. Right? And that's where plant roots tend to be. So this is really important. Say you're a farmer, or if you go to the store, you could actually buy topsoil. It's because that's an important aspect of the soil. It's an important layer of the soil, especially if you're trying to grow something. That's where the roots are going to go. Some soils have an alluviation layer here, and that has to do with the way ions move in and out of the soil, but we're not really going to get into that because it's not relevant to this course. And then below that, you get into just more rocky stuff, not a whole lot of nutrients. The further down you go, the more like rock stuff is, the less organic matter, the less accessible nutrients for the plants. Okay. So just a quick aside, out in the Great Plains, okay, you guys have heard of the Dust Bowl? Okay. So in the Great Plains, they were grasslands before we had a mass migration of people out to the Great Plains. They were largely grasslands. You know, buffalo out there, plains animals. The problem with them was that when the farmers farmed them, the way they farmed them, it disrupted the fragile ecosystem such that the topsoil dried up and blew away. Okay? And it rendered huge amounts of land relatively infertile. And this is basically all sort of dried up topsoil just whoosh, blowing away. The woman in that picture, I think, is 30, which is kind of absurd because she looks much older. Okay. So how do plants get these nutrients that are in that soil? Well, it turns out that with negatively charged ions, so any ion this plant needs that has a negative charge, it's pretty easy for it to get from the soil because the soil itself, soil particles themselves, for whatever reason, tend to have a slight negative charge. Negative repels, so it's pretty easy for the plant to gain access to those negatively charged ions. In terms of positively charged ions, it's a little harder because those positively charged ions stick to the slight negative soil. Okay, so you guys ever watched Indiana Jones, an old Indiana Jones movie? Okay, well, so there's this great scene where he's got to take this stone idol off of like a thing, and he knows that if he takes it off, right, to trigger some mechanism, so he has to replace it with something that weighs exactly the same.
Yeah.
Right, but he triggers it anyway with a giant ball, chases him on it. Okay, so anyway, the basic idea is that plants do sort of the same thing. Okay, they try to pull a bait and switch on the soil. They pump out hydrogen ions. Remember, they have those proton pumps. What do proton pumps use? What do proton pumps need?
Oh, ATP, right?
ATP, yeah, they need ATP, right? They actively pump out hydrogen ions, and it turns out the hydrogen ions pop these positively charged, say, potassium ions in this case, off the soil and allow the plant to take it. Okay, so the plant has a sneaky way of getting around the fact that positively charged ions are bound to the soil and hard to get at. Okay. All right. So, in terms of major nutrients, the only one I'm going to spend a lot of time talking about is nitrogen. Carbon, the plant gets from the air, oxygen, the plant gets from the air. A lot of the other nutrients, the plant gets from the soil. In terms of big nutrients the plant needs, one of the most interesting ones is nitrogen, because the plant can't use nitrogen from the air. Atmosphere is about 80% nitrogen. The plant can't use any of it. Neither can we. Okay, so the way plants get around that is there are bacteria that live in the dirt. Two particular kinds that are interesting if you're a plant. One and that decaying material into ammonia, which is something that the plant, at least in small quantities, can use. Okay, so the plant can use nitrogen in the form of the ammonium ion, at least in small quantities. The other kind of interesting bacteria is nitrogen-fixing bacteria. Okay, so it's not that the nitrogen is broke. Okay, nitrogen's not broken. It's that this bacteria can take atmospheric nitrogen and turn it into ammonia. Okay, ammonia is a more accessible form if you're a plant. Plants also like this third kind of bacteria, nitrifying bacteria. Nitrifying bacteria takes ammonia, turns it into nitrate. Nitrate is the major form of nitrogen that plants use. Okay, so plants need bacteria. Without bacteria, plants wouldn't have access to nitrogen and plants would not live. Okay? We, therefore, need bacteria because without plants we wouldn't live, and without bacteria, the plants wouldn't live. Does that make sense? Okay, so plants actually, some plants have this really cool, symbiosis. So any of you guys do any sort of farming or gardening? Okay. Do you guys know what crop rotation is? Have you ever heard that phrase?
Yeah.
So, okay. Yeah, so crop rotation is you don't grow the same thing year after year in the same soil because then you drain the same nutrients out. Different kinds of crops have slightly different nutritional requirements. It's not like, the bean plants need exactly the same thing as potato plants need exactly the same thing as corn plants. All need slightly different arrays of nutrients. So you don't ever want to grow the same thing year after year after year because you crap your soil. Soil's no good. One kind of cool thing farmers will do is they'll grow like a harvest crop, corn or something, and then the next year they'll plant a legume. Legume beans. Okay? Because legumes have this cool symbiosis with the bacteria where the plant recruits, calls in, sends out a chemical signal that calls in bacteria. The bacteria live in nodules, live in these growths of the fruit. So the bacteria get this nice sheltered place to live. Some of these bacteria are anaerobic, all these other things. They need some protection. They get that protection from the plant. In return, these bacteria fix a ton of nitrogen. They make a ton of nitrogen available for the plant. In fact, they make so much nitrogen available that by growing a legume in your field, you actually render your field more fertile next year. You actually are basically fertilizing the field by growing beans. These bacteria add nitrogen to the soil. Not only do they give it to the plant, but they give some back to the soil. Okay. Human beings thought it was awesome how nitrogen fixation could work. So human beings investigated the processes by which bacteria fix nitrogen, take nitrogen from the atmosphere and turn it into nitrogen. And we came up with something called the Haber process, which requires all this elaborate machinery. Now, the problem with the Haber process is it's kind of too good. Okay, so you make nitrogen-based fertilizer, you ship it to the farms out in the Great Plains states, the states that are up close against the Mississippi River. Okay? Farmers put all this awesome fertilizer on their fields. It's nitrogen-rich fertilizer made by taking atmospheric nitrogen and turning it into nitrates. It's great for your crops, but when it rains, okay, a lot of those nitrates wash in the Mississippi River. Okay, so we've got nitrogen in the Mississippi River. Still, what's the big deal? All that nitrogen washes down to the Gulf of Mexico. You know what the dead zone is? Have you ever heard of this? Okay, not the Stephen King novel. Okay, so it washes down into the Gulf of Mexico. In the Gulf of Mexico, there's this huge swath of water where there's no fish, nothing, not one, nada, nothing, and it's been very big in certain years. Like I think it's the size of Texas, maybe even bigger than that. Like we're talking massive amount of water where there's no fish. So why is that? Why do we go from nice fertilizer for your field to no fish? Okay, here's what happens. So all that fertilizer rushes into the Gulf of Mexico. It's plant fertilizer. Algae have a lot of the same nutritional requirements as plants. The algae go bananas. Okay, so still, what's the problem? So we got a lot of algae. The problem is the algae dies, and as it dies, it settles. As it's settling, there's bacteria in the water that decay it, break it down. And those bacteria are aerobic, they need oxygen. So as that algae is settling down and being decayed, the bacteria are sucking all the oxygen out of the water. So there's so little dissolved oxygen in that part of the Gulf of Mexico that fish cannot live. They die. Okay, it's called it's the eutrophication process.
So are there any fish in Mississippi?
Yeah, because it's not until so the water's kind of moving there, right? It's coming from a source moving through, taking up nitrogen, and then building up when it gets to the Gulf of Mexico. It's the same phenomenon we used to see with similar phenomenon we used to see with kind of like bald eagles. So you guys know why the bald eagles almost went extinct? One of the reasons.
DDT.
Yeah. So DDT got in a fish. It wasn't a lot that got into the fish. So the fish, what got into insects, got into the fish. Not so much in the fish. So the fish were fine. They were pretty healthy because they only had small amounts. When the eagles ate a lot of fish, all those fish with that DDT built up in the eagles and it caused their eggshells to crack. So same thing here.
What's
DDT is an insecticide. In fact, there was an interesting, when I was still in grad school, I remember seeing people petitioning to re-legalize DDT because it's an effective insecticide for mosquitoes. So there was kind of a debate about, okay, we could kill off malaria-carrying mosquitoes was their intention. So isn't that more important than birds? And it was an interesting intellectual discussion. It turned out DDT wasn't really the solution. But still, it was an interesting intellectual exercise. So what do? We kill all the insects, malaria, or do we worry about how many species of birds we're just going to knock out? Okay, so same idea though. As the Mississippi is coming through those Great Plains, the water at any given point doesn't have a ton of nitrate in it. When it empties out in the Gulf of Mexico though, we've got all that nitrogen that was brought there 5 minutes ago, 10 minutes ago, an hour ago, and it's all right there. And so you just get a really high concentration of nitrates there. That's why it's one of the reasons why they have the fountains going in the pond. Without the fountains going in the pond, you get a nice scum of algae. The algae would settle, the water would become deoxygenated, and all the fish in the pond. Okay. So nitrogen, we've found a way to sort of make use of nitrogen fixation, but we're not particularly careful about the way we use it, and that's had some interesting, pretty awful unintended consequences. Okay, so nitrogen fixing bacteria in legumes is one symbiosis plants have. Another one that we already talked about is mycorrhizae. Okay, there are two kinds. Ectomycorrhizae, which put their hyphae through the apoplast. Remember the apoplast? What's the apoplast? It's it's sort of like going through their cell walls. It's usually the cell wall. So yeah, the apoplast is going through here without ever entering the plant. There are endomycorrhizae, which put their hyphae kind of into the plant cells. They actually put it through the cell wall, and then they kind of poke into the membrane. They don't actually go through the membrane. They poke kind of an indentation into the cell membrane. Both ecto and endomycorrhizae work about the same way. They get nutrition from the plants by sticking their hyphae in, but they bring water to the plant, so they help increase the surface area of the roots. Okay. Everybody good so far? Okay. All right, so that's pretty much it for the plant nutrition chapter. There's not a whole lot in there. They go into like what a plant looks like if it doesn't have enough magnesium. That's not particularly, I don't think that's helpful for the AP exam, nor is it particularly interesting, so we're just going to skip it. But there are some really cool adaptations plants have for variations from the normal get your nutrients from the ground, make sugar from your leaves routine. For example, this is something called an epiphyte, okay? And you can see it's way high up off the ground. An epiphyte is a plant. This is a bromeliad.
It looks like a Dr. Seuss book.
It does, doesn't it? Yeah. Bromeliads just put their roots down into, say, a hollow of the tree where there might be some water collected. Okay, so these things don't grow in the soil. They actually grow way high up on other plants, on trees. Okay, so that's an epiphyte. This is something called dodder, which is a parasitic plant. So this kind of haze of orangey stuff is actually a plant that has no chlorophyll because it doesn't do photosynthesis. Instead, it's entirely parasitic. It sticks its stem into another plant and you've ever grown chrysanthemum, it's common to have daughter on the chrysanthemum, and they put out the wild rose. These are carnivorous plants. We're going to actually watch David Attenborough mess with some carnivorous plants.
Animals don't eat it. It eats animals. And there's one right here. Watch.
I want to be like this when I'm 60.
This is Venus's flytrap. It shapes its traps from the ends of its leaves. One or two hairs on their surface act as triggers. Here comes a meal.
Oh no, turn away! No, don't go into the light!
Touch the hair, and the trap is sprung. There's now no escape. The beetle's struggles stimulate the plant to close the trap even more tightly. It now produces digestive acids from glands on the inner surface of the leaf, which first kill and then dissolve its victim's body. Growing in the same Carolina swamp, there's another carnivorous plant. These are the trumpet pigeons. They, like the Venus's flytrap, find so little nutriment in this impoverished, waterlogged soil that they supplement it with the bodies of animals.
So you could find pitcher plants around here, not that big, but there are pitcher plants.
Their traps are also formed from leaves, but leaves that have been folded lengthways to make a vertical tube which fills with water. These spectacular trumpets may look like flowers, but of course they're not. Though, in a sense, this bright yellow top to them serves the same purpose as a petal. It's an advertisement of a delicious reward, and the reward itself is under here. Sweet nectar. But if an insect comes to collect it and strays into the mouth of the trumpet, then it's doomed. The inside of the throat of the trumpet is covered with microscopic downward pointing spines. As long as it stays on the rim, the ant is all right. But if it strays off it, it falls into a pond of water and drowns. The tiny corpse dissolves, and the marsh pitcher absorbs the resulting soup. Where one ant goes, others are likely to follow.
It's got little hairs on it, so it's really slippery. So if they step in the wrong place, they just slide right off.
The marsh pitcher attracts other animals too. This frog may be hoping to eat some of the insects before the pitcher does. But if it loses its footing, the plant will eat it.
Actually though,
Yeah. Yeah, well, so, I mean, the plant produces digestive enzymes that will digest whatever drowns in that water. Of what? Yeah. Oh. David, well, I showed you when we get to, I didn't I show you the bird pond when they do the birds, where they do all the fancy dances, and it's spectacular.
oh yeah.
And the bird's like,
Oh
I
yeah.
was like, ah!
Oh,
Yeah, David Attenborough is by far. That's why we watched the Life videos, like after he did them. Not Life, Planet Earth. Because he did them. What? Yeah, he does those. I remember watching him when I was in AP Bio and like going, that's what I want to be my girl. That's what I want to be. Because he's like doing all these crazy things. Like the best one I remember, he's coming out of the ocean in like full on scuba gear. And he's, gotta be in his 50s, 60s. And he's like talking, but he's still got the regulator on, so it's like, it's just like, you could take the thing out of your mouth and talk. He is my hero.
Big deal, man.
Yeah.
All right, so just as a foreshadowing, next week when you guys, for you guys, I probably won't do it on Friday. It's going to happen on H block for everybody else. For you guys, I'll probably do it the day you have a super because I don't want to wait till Friday. We're going to do a quick lab where I'll set most of it up. I'll have you come in prepared to do it. We'll do lecture for half the class and then you'll take measurements for the other half. So it'll be a really quick lab. Okay, so I'll send that out. Probably look for an email over the weekend with the assignment in case you want to get a jump on it. But it won't be due, I have to figure out, probably not until after the plant test. Okay, I don't know if you saw the email, but the plant test will be a week from next Monday. Because we're going through it pretty quick and in not a lot of detail. So we should be well done by next Monday. I mean, I have a feeling we'll be into ecology before the test. Okay? Everybody good so far?
So is the test a week from next Monday?
It's a week from this coming Monday, yeah. So a little over a week. Okay. Alright, so just because you guys especially, a lot of you guys didn't take advance, we're going to be talking about something that's new to you guys, alright? And that is this idea of alternation of generations. Okay, because the next chapter on plants is about plant reproduction. And so this chapter we're actually slow it down and spend a little more time on because it's somewhat more complex. Well, it's a lot more complex than the nutrition chapter. Okay, so the first thing you have to understand, if you haven't taken advance or if you've forgotten, is this way that plants do reproduction that's kind of funky. Plants have two different multicellular life stages. And we have talked about this a little bit when we talked about the differences between the fungal reproductive cycle, the plant reproductive cycle, and the animal reproductive cycle. So we've already alluded to this, but here's the concept laid out in a little more detail. In plants, you have a diploid plant, okay, that is called a sporophyte. The sporophyte has cells, reproductive cells, that undergo meiosis to produce haploid spores. The haploid spores grow by mitosis, so they get bigger, to produce a haploid plant called a gametophyte. And I know a lot of this isn't in your notes, and the reason for that is that until this year, I've had very few kids who haven't had this. Right? This is the first year where I've got 20 of you who are upper mids. The most I've ever had before was 2. Okay, so that's why this isn't in your notes, because I haven't had a lecture on this too much. Okay, so gametophytes are the multicellular haploid stage of a plant. Via mitosis, because they're already haploid, via mitosis, regular cell division, gametophytes make haploid gametes. Gametes undergo fertilization. Okay, a male gamete and a female gamete fuse to form a fertile zygote, which is diploid. Okay, and the cycle repeats. The zygote grows into a multicellular diploid plant. The diploid plant makes haploid spores. The haploid spores grow into multicellular haploid plants, which make haploid gametes, which give rise to diploid sporophytes. Okay, so why, when you look out the window, don't you see 2 versions of a pine tree, 2 versions of a dandelion, 2 versions of an oak tree? Well, it turns out that in most of the plants that you guys are familiar with, this is the only one, sporophyte is the only thing you can see. Okay? So in the angiosperms and gymnosperms that you guys are most familiar with, this is what you're looking at. You're looking at a diploid plant that makes spores. In the plants that you are used to looking at, the male gametophyte is only a pollen grain. That's it. That's the whole male sexual plant. It's a pollen grain. Okay, and it's really, as an immature plant, it's 2 talking about something that's very reduced. Okay, the female gametophyte you can't even see it. It's contained entirely within the female part of the plant, the carpel. Okay, it's contained entirely within the base. There's a swollen base on the carpel. It's contained within that, entirely within that swollen base. Okay, so there's a reason why you don't look out the window and see two versions of a daffodil plant, two versions of the plant. Because one of them is very small. It's multicellular. The gametophytes are multicellular, but they're teeny, they're tiny. Pollen grains, and then the female one is actually tucked inside a sporophyte. It never leaves the sporophyte. Okay, now, more ancient plants, like mosses and ferns, if you were to look very closely on the forest floor, you might see little plants that look like this, that are the gametophytes of a fern. Okay, the gametophytes of a fern. Okay, and moss is actually the thing that you see, the nice fuzzy soft stuff. That's actually the gametophyte, the sporophytes, if you've ever seen them. So here's the fuzzy, nice moss stuff. The sporophytes stick up over it and look kind of like that. Okay, so in more ancient plants, there are two macroscopic generations. The gametophyte generation and the sporophyte are both visible to the naked eye.
Why is it just ancient?
It's just the way evolution has happened over evolutionary time. The gametophytes have become smaller and smaller and smaller and smaller and smaller, so that in the plants that evolved the most recently, at least according to evolutionary theories, the plants that evolved the most recently have the smallest gametophytes. I mean, some of the advantage to that is, okay, so where do you find moss growing? Do you find it growing in a place that's very dry? No, they grow in damp places. The reason for that is that the sperm have to actually swim through water to get to the female gametophyte. Okay, so the male gametophyte releases little sperm that swim through the water. You know, we're not living in a uniformly damp environment. The terrestrial environment is quite dry in some places. So having the male gametophyte being a little tiny airborne piece of pollen, or pollen that can be carried by an insect, is actually quite useful if you want to get the sperm from the egg without having to worry about swimming through water. Okay, fern as well. Ferns are another one where the gametophyte is actually macroscopic. It's small. It's also close to the ground. You tend to find fern in wetter places. And the reason these are close to the ground is that way the sperm can swim through the water. All right, everybody good? Okay. All right, so here's another depiction of alternation generations with some more terminology. So you have the diploid sporophyte. Meiosis of certain cells in that sporophyte produce spores to make the female gametophyte. You start with a cell called a megasporocyte. Megasporocyte goes through meiosis to produce spores. The spores are called megaspores. Megaspores give rise to the female gametophyte. If you're talking about the precursor to a male gametophyte, it comes from something called a microsporocyte. Okay. Microsporocytes go through meiosis to produce microspores. Microspores give rise to male gametophytes. So spores give rise to the haploid gametophytes, which in most of the plants you guys are familiar with, you can't see very well. I mean, sometimes, if it's really bad out, in a couple of weeks, you'll see pollen blowing through the air. That's pretty much it. Other than that, these gametophytes are largely invisible. Two gametes, a male and a female gamete, come together in fertilization to produce a zygote. Zygote develops into a plant embryo, a seed. Plant embryo grows up into what you guys are all familiar with, the giant diploid sporophytes. Okay. So, all right, let's keep going just a little bit and then we'll dissect some flowers. So just to overlay now even more terminology, and this is the picture I've given you and I've put in some blanks for you guys to fill in. Okay, I didn't give you exactly this picture because I couldn't find exactly this picture. this is how flowering plants work. Okay. Within the reproductive parts of a plant, okay, we have the male part of the flower and the female part of the flower. Now, not all flowers are perfect flowers. Perfect flowers, male and female parts. Not all plants have both male and female parts. Male and female parts. Can you guys know plants that's fairly common? Well, it's less common up here. It's an ornamental plant that you have to buy separate male and female, or a tree that just cut down outside of town. I don't know. I don't think so. I think most of our kids are.
I remember the tree that was by the grandma.
Yeah. The ginkgo. It's a ginkgo tree. Yeah. So ginkgo trees, there's two different gender trees. There are trees that make flowers that only have female parts, and there are trees that make flowers that only have male reproductive parts. And holly is a holly, you can male and female plants. And then there's some really weird plants. Like we grew this one when I lived at Chicopee. We had this vine. I can't remember the species, but it was this really weird vine that had two different flowers on it. And the flowers looked totally different. One of the flowers had only female reproductive parts. The other flower had only male reproductive parts. It was very strange. That's not really common. I can't remember the name of the vine, but it was really cool vine. Okay, so here we're just going to look at the most prevalent situation where you have both male and female parts in the same flower. Okay, so we've got the anther, the male part of the flower, which produces the male gametophytes containing these pollen grains. Pollen grains land on the female part of the flower, the carpel. The pollen grain germinates, okay, it grows, sends down, this is one long cell called the tube cell, sends down this really long cell, and then down the tube cell swim two little sperm. Down here in the ovary of the plant, okay, so this big area here is called the ovary. Down here in the ovary of the plant, we have a container called an ovule. Within the ovule is the female gametophyte. Okay, so the female gametophyte never leaves the sporophyte. The female gametophyte is entirely contained within the sporophyte. Okay, so pollen grain comes down, grows down, two little sperms swim down, they grow through a hole here called the micropyle. Sperm go through here into the area where the female gametophyte is. Okay, the female gametophyte, as we'll see, is composed of several different cells. The sperm fertilize two of those cells, and we'll talk about how that happens later. Okay, a seed develops, grows into a mature sporophyte, and the process repeats again. The sporophyte makes spores, two different kinds of spores, microspores and megaspores. Megaspores grow into female gametophytes. Microspores grow into male gametophytes. All right, so what I'm going to have you guys do now, I'm just going to play with some flowers. And I know they're dead, but I got them for free because they're dead. So you're going to open them up. And we're actually going to go in the back and get some dissecting microscopes. Oops, sorry. And they're kind of icky, but the important parts are still intact. So check to make sure. All right, so these are daffodils. Daffodils have an interesting floral structure. Okay, so you know how daffodils look kind of like a trumpet? Okay, let's spread it up. This was closed, but I could probably read them. All right, so daffodils have an interesting structure. They have this kind of trumpet here. This is made up of fused petals. All right, and then this part here, this is actually a sepal. If you've ever seen like a carnation, right? When a carnation is about to blossom, the carnation has those green pieces. Okay, in most plants, or most flowers, the sepals are green. Bless you. In daffodils, the sepals are modified so they actually look like petals. But this is the petal of daffodil. Inside, you're going to see the female reproductive part called the carpel, and male reproductive part called the stamen. Alright, so what we're going to do, okay, so here's what our flower looks like. There should be the carpel, which consists of the sticky stigma where pollen sticks to, the style, which is just a long tube, and the ovary, which is where the female gametophytes come from. The male part consists of a filament, which holds the anther up in the air, making it more accessible either to wind or to insects. The anther, which is where the pollen grains are produced. Okay? And then we have the protective parts, or the parts designed to attract insects. The sepals, which are largely protective, and then the petals, which are often designed to attract pollinators. They're either the flower that produces a scent, or the petals are a bright color, or in some cases, the petals actually have ultraviolet landing strips. I swear to God, ultraviolet landing strips marked on them. In some cases, they do like here. In the best one though is, I'm going to show you this later maybe, is another David Attenborough clip where he's got an orchid that is designed to look like a wasp. So wasps try to mate with it, I swear. The wasps are like trying to hump this flower, and they pick up pollen when they're like, you know, trying to get busy with the flower. Okay, little do they know. All right, so what we're going to do now is just so you get a really good look at this, the piece of the flower. I mean, so get some experience with a different kind of microscope. You guys are going to come in here. Oh, you know, honestly, okay, what we're looking for, as long as they're still back here. You guys are going to play with dissecting microscopes today. They look like this. Okay? So note that there's no lens to swivel around, and there's two eyepieces. So go back, and I guess two people to it. There's not a whole lot of scopes back there, so get two people to a scope. And then you can play with your flower and see what you can see underneath the scope. You know what? So I got one for somebody who wants to be lazy and have her back there. There's two lights. You want to turn. You guys want to turn on the one that comes from above. You want to turn off the one that comes from below. You want to want that. You want the one that comes from above. Okay? And so you're just gonna get your fingers. Oh, you have on that. Yeah, you want the one coming from above, which is on. He wants to share my life.
Jerry, did you keep saying your Our kid's not going back there.
Yeah, you're just gonna open it up.